reproducing, but a species cannot survive without reproduction. Reproduction is thus essential for the continuation of species. It ensures that the genetic material of one generation is transmitted to the next. Each generation produces more offspring for the next generation. Many individuals die due to various reasons like disease, competition, genetic factors, etc. Before reaching the reproductive age, only the fittest and the best survive and reach the reproductive age. This ensures that the advantageous characteristics are transmitted to the next generation. Asexual reproduction means simple cell division that produces an exact duplicate of an organism. There are many types of asexual reproduction. Sexual reproduction involves the joining fusion of male and female sex cells, gametes. Methods of asexual reproduction. Asexual reproduction does not involve the fusion of gametes. There are many types of asexual reproduction, all producing individuals that are genetically identical to each other and to the parent. Binary fission. Binary fission means division into two. It is the simplest and most common method of asexual reproduction. It occurs in prokaryotes, bacteria, many unicellular eukaryotes, for example, protozoa and some invertebrates. During binary fission in bacteria, the DNA is duplicated and so two copies of DNA are formed. The two copies move towards the opposite poles of cell. The cell membrane invaginates the in center and divides into cytoplasm into two. New cell wall is deposited between two cross membranes. It results in the formation of two daughter bacteria which grow in size and divide again. During binary fission in unicellular eukaryotes, the nucleus of parent organism divided into two by mitosis. It is followed by the division of cytoplasm, so two daughter cells of almost equal size are formed. Daughter cells grow in size and then divide again. Some invertebrates also reproduce asexually through binary fission. During this reproduction, body is cut into two halves fission and the missing body parts are regenerated in both halves. This type of asexual reproduction is common in Plantaria and many ecoderms. Fragmentation. As certain worms grow to full size, they spontaneously break up into 8 or 9 pieces. Each piece fragment develops into a mature worm and the process is repeated. If a planarian breaks into many pieces instead of two, it will also called as a fragmentation. Budding. In this type of asexual reproduction, a bud develops as a small outgrowth on parent's body. In case of yeast and unicellular fungus, a small bud is formed on one side of cell. The nucleus of cell divides into one daughter nuclei and passed into the bud. Parent cell may form more than one bud at a time. Each bud enlarges and develops the characteristics of parent's organism. The bud may separate from parent body. In some cases, the bud never separates and as a result, colonies of individuals are formed. Animals such as sponges, hydra, and corals are reproduced by means of budding. In them, a small bud is formed on the side of the body by mitosis. The bud enlarges by the formation of more cells. It is then detaches from the parent body and grows into new organism. In corals, the bud do not detach from the parent body. Corals form big colonies because the bud grows into new organisms by remaining attached to the parent body. Spore formation. It is generally seen in most fungi. When Rhizopus reaches reproductive age, its body cells from thick walled spore sacs called sporangia sing sporangium. Inside each sporangium, a cell divides many times and forms many daughter cells called spores. Each spore is divided with a thick wall called cyst, and it can survive unfavorable conditions. When sporangia are mature, they burst and release spores. Under favorable conditions, the spores germinate and develop into new rhizopus. Under unfavorable conditions, some species of bacteria reproduce by forming spores, for example, Bacillus species. The bacterial spores are also thick-walled. They are formed inside bacterial cells, so are called endospores. Um, Parthenogenesis, parthenogenesis is also considered as a form of asexual reproduction. In it, an unfertilized egg develops into a new offspring. Some fishes, frogs, and insects reproduce by meaning of parthenogenesis. Similarly, queen honeybee lays eggs in the cells of a honeycomb. Many eggs remain unfertilized and develop into haploid males drawn by parthenogenesis. At the first time, some eggs are fertilized by male bees, and these develop into diploid families, new queen and worker bees. Vegetative propagation. When vegetative parts of plants, i.e., roots, stems, or leaves, give rise to new plants, the process is called vegetative reproduction or vegetative propagation. It occurs naturally and can also be brought about artificially. Natural vegetative propagation occurs naturally in several ways. Bulbs are short underground stems surrounded by thick fleshy leaves and that contain stored food. Adventitious roots emerge under the base of bulb while shoots emerge from the top of base. Tulips, onions, and lilies reproduced by bulbs. Corms are short and swollen underground stems containing stored food. Buds are present at the top of corn. From a bud, shoots grow from and new plant. Daishin and garlic reproduced by corns. Rhizomes are horizontal underground stems with scale leaves. There are enlarged portions called nodes on rhizome. Buds are produced at nodes. The bud present on the upper surface of rhizome give rises to shoot. The lower surface of rhizome produces adventitious roots. Ginger ferns and water lilies reproduced by rhizomes. Stem tubers are the enlarged portion of an underground stem rhizome. There are aggregations of tiny buds in the form of eyes along the surface of tuber. Each bud develops into shoot that grows upward and also produces roots. Potatoes and yams reproduced by tubers. Suckers are lateral stems close to ground level. A sucker grows underground from some distance and then turns up, producing the new plant mint and reproduced in this way. Vegetative propagation by leaves is not common and is seen in plants by as Pathier chut. This plant has fleshy leaves and adventitious buds are present at the margins of leaves. When leaf falls on ground, the bud grow into new plants. Artificial vegetative propagation. Gardeners and farmers use artificial methods of vegetative propagation to increase the stock of a plant. The following are most common method of artificial vegetative propagation. Cutting. In this method, cutting may be taken mainly from the stems or roots of parent plant. These cutting must be have meristematic region from which growth can occur. When cutting are placed in a suitable soil and under right conditions, sufficient nutrients, water, and sunlight, they from roots and shoots. Roots and shoots grow up and develop into a plant identical to the parent plant from which the cuttings were taken. Roses, ivy, and grape vines are propagated by stem cutting. Sweet potato is an enlarged root. Farmers place it in moist sand or soil until it produces several plantets. Then the plantets are removed and planted. This process is used to produce many plants from a single plant. All new plants are exactly the same. This artificial vegetative propagation has been very beneficial on sugarcane plantation. Grafting. In grafting, a piece of stem is cut from the plant and is attached with another plant with established root system. After a while, the vascular bundles of attached system piece and the host plants are connected to each other. The stem piece and the plant begin to grow together. This method is used to propagate many roses, peach trees, plum trees, and various seedless fruits, including grapes. Advantage and disadvantage of vegetative propagation of plants. Plants can reproduce asexually via vegetative propagation. This method of reproduction has some advantages and disadvantages as well. Advantages: the offspring produced through vegetative propagation and genetically identical, therefore beneficial characteristics can be preserved. In vegetative propagation, there is no need of any mechanism of pollination. It helps to increase number of plants at a rapid rate. The organ of vegetative propagation enables many plants to pass over unfavorable conditions. Plants bearing seedless fruits can be grown only by vegetative propagation. Disadvantage is the plant do not have genetic variation. Species specific diseases can attack and this can result in the destruction of an entire crop. Tissue culture and cloning. Cloning is the latest method of vegetative propagation. In this method, identical offspring are produced from a single parent using its vegetative tissue or cell. Tissue culture is the technique applied in this method. Tissues are taken from any part of a plant and are put in a suitable nutrient medium. The tissue cells start mitosis and produce masses of cells called celluses are transferred to other medium that contains different hormones for the formation of roots, stem, and leaves. Calyuses make these structures and grow into new small plants. The small plants are then planted into pots and then in fields. Sexual reproduction in plants. Sexual reproduction involves the production of gametes, sperm, and egg cells and their fusion fertilization. Gametes are produced in special structures in plant body. The major plant groups are mosses, ferns, and seeds. The seed plants include gymnosperms and Angiosperms. Flowering plants. Plant group are used different methods for bringing the sperm and egg cells together. In mosses and ferns, sperms are motile and swim to egg cells. Therefore, these plants require water in the form of dew or rain for sexual reproduction. In the other hand, angiosperms have special methods for carrying their sperm to egg cells. They do not need water for reproduction. In the life cycle of plants, two different generations alternate with each other. One generation is diploid and produces spores. It is called sporophyte generation. The other generation is haploid and produces gametes. It is called gametophyte generation. In the phenomenon in which two different generations alternate with each other during life cycle is known as alternation of generations. In most plants, sporophyte generation is dominant. It means that it is bigger in size and it is independent. Sporophyte produces haploid spores by meiosis. The spores develop into gametophyte. It is small in size and develops on sporophyte. It produces gametes by mitosis. The male and female gametes fuses and form diploid zygote. The zygote undergoes repeated mitosis and develops into a new diploid sporophyte. Sexual reproduction in flowering plant. We know that in angiosperm, parent plant is diploid sporophyte generation. Flower is the reproductive structure in this generation. The flower components are arranged in the form of whorls. The outer two whorls in a flower are the non-reproductive whorls, while the inner two whorls are the reproductive whorls. Calyx. Calyx is the outermost whorl. It is usually green in color. Its individual units, leaflets, are called sepals. Sepals protect the inner whorls at birth stage. Corolla is the next inner whorl and is often colored brightly. Its individual units, leaflets, are called petals. They serve to attract bees, birds, etc., which are the agents of pollination. The flower is actually a condensed shoot, while the nodes with the nodes present very close to each other. The different parts of the flower are attached to the nodes. All the structures present at the node are collectively called the whorl. The third whorl and the androecium is the male reproductive part of flower. Its units are called stamens. Each stamen has a thread-like filament at the free end of which anther is attached. Anther has pollen sacs in which haploid microspores, pollen grains, are produced through meiosis. Each microspore germinates into the male gametophyte during generation. During it, the nucleus of microscope undergoes mitosis and produces new nuclei. A tube nucleus and generated nucleus. The generated nucleus again undergoes mitosis and produces two sperms. So a germinated microspore has a tube nucleus of two sperms. All these structures are male gametophyte generation of plant. Fourth whorl, gynoecium, is the female reproductive part of flower. Its units are called carpels or pistils. Each carpel is made up of the base, basal, ovary, and middle style and upper stigma. Inside ovary there are one to many ovules. Inside each ovule the haploid macrospore is produced through meiosis. Microspore germinates into the female gametophyte generation. During that, macrospore undergoes mitosis and produces an egg cell and some associated structures, e.g. fusion nucleus. Egg cell and associated structures are the female gametophyte generation of plant. Different flowers have one to many carpels. These may be either to fuse to free. When pollen grains mature, they are transferred to stigma. It is called pollination. On reaching the stigma, the tube nucleus of pollen grains construct a pollen tube. The pollen tube contains a tube nucleus and two sperm. The tube grows through style and ovary and enters ovule. Here it bursts that releases the sperm. Both sperms enter the female gametophyte. One sperm fuses with eggs and forms a diploid zygote. Diploid zygote sperm fuses and the other sperm fuses with diploid fusion nucleus and forms a triploid 3N nucleus called endosperm nucleus. Since the process of fertilization involves tube fusion, it is called double fertilization. Zygote develops into embryo and endosperm nucleus develops into endosperm tissue, food of the growing embryo. Ovule then becomes seeds and ovary change into fruit. When seeds mature, they are dispersed. If seeds get suitable condition, their embryos develop into new plants, the haploid sporophytes of the next generation. Pollination is defined as the transfer of pollen grains from flowers another to stigma. Two types of pollination are recognized. Self-pollination defined as the transfer of pollen grains from the anther to the stigma of the same flower or other flower of the same plant. Cross-pollination is transfer of pollen grains from the flower on one plant of the flower on other plant of the same species. Cross-pollination is brought about by various agencies like wind, water, bees, birds, bats, and other animals, including man. The insect-pollinated and wind-pollinated flowers have structural adaptations that facilitate the transfer of pollen grains between two plants. Examples of insect-pollinated flowers are buttercup, rose, wallflower, sunflower, orchid, etc. Examples of wind-pollinated flowers are grasses, hazel, willow, corn, etc. Development and structure of seed. We know that after fertilization, the female gametophyte zygote divides repeatedly by mitosis and develops into an embryo. At this stage, ovule changes into seed. The formation of seed completes the process of sexual reproduction in seed plants. Angiosperm seed consists of three distinct parts: the embryo formed from zygote, the angiosperm tissue formed from endosperm nucleus, and the seed coat which develops from the wall of ovule. Seed coat develops from integument originally surrounding the ovule. It may be a paper-thin layer or thick and hard coconut. Seed coat protects embryo from mechanical injury and from dying out. There is a scar on seed coat called helium. It is where the seed is attached to ovary wall fruit. At one end of helium there is a micropyle. This is the same opening through which the pollen tube entered ovule. The seed